What do you say, Vasha?
Rolf, can you hear me?
I can hear you.
Yeah, you can start now.
Alrighty. I'll stop the camera so you don't have to stare at my face. Okay. Good morning, everybody. This is the, let's see, is it working? No. No, it's working. Good morning and welcome to the Vertebrate Limb Development lecture with me. And I took almost all the information to make this lecture from two sources. It's the 18th edition of the Developmental Biology book and the latest review I found on limb development, which came out just a few months ago by Kathy McQueen and Matthew Towers. And here are the references. Okay, the outline of the lecture is I'll start with an introduction. I'll go through the, just show you the signaling pathways, which will reoccur over and over again. I'll talk a little bit about Hox genes and the limb bud, because it's kind of important part of this lecture. Then we'll go over to general anatomy, and we'll talk about limb fields, which you will understand what that is soon. Limb bud initiation, axis formation, and we have three axes, of course. We have the proximal-distal, we have the anterior-posterior, and part of that is then the digit identity, and then the dorsal and ventral axis. Okay, let's jump straight in. So these are the main signaling pathways. And by now, you have, of course, heard of these over and over again, but I thought I'll just repeat in case some of you have forgotten. We have the Wnt pathway, which is named from the Drosophila wingless and INT1, which is a family of secreted ligands, which, as you already know, have multiple functions during development. We have the fibroblast growth factors, which are also secreted ligands, which, as you know, have multiple roles during development. We have sonic hedgehog, which in this context we treat as a secreted ligand, and I'm sure you've heard of this before because it takes part in multiple developmental processes. We have the bone morphogenetic proteins, which are part of the transforming growth factor beta family, which are secreted ligands, and they are quite common in developmental processes. And finally, we have the almost ubiquitous retinoic acid, which in the context of limb development will be treated as a secreted ligand. And I'm sure you've heard of this before. It's pretty much everywhere in development. Now, Hox genes, I know that you heard about Hox genes in your flight talks. These are homeobox transcription factors. And in Drosophila, there are segment identity genes, and they can have this function as well during vertebrate development. Now, tetrapods have 4 clusters, the A, B, C, and D, with a maximum of 13 genes in each cluster, and it varies between the species. Now, what's interesting with them is that they have a spatial and temporal collinearity of expression. That means that the 3' genes, which is the 1, 2, 3, and so on, they will be expressed first. And the 5' genes, which then will be like 11, 12, 13, will be expressed last. And they are involved in things like specification of axial identity, and as you will hear today, limb development. And this is then a generalized vertebrate, and you see that there is a nested expression in the CNS. You have in the head, the most anterior ones are the ones with the lowest numbers. So we have 1 and 2 coming here, and so on and so forth, and you have higher number the further posterior you go. And you can find a similar expression also in the paraxial mesoderm. So this is expression in the somites. And of course, which they don't show here, it's the same in the lateral plate mesoderm. Well, it's not the same. It also has a nested pattern. Okay, this is the summary of limb developments. So the lecture today will essentially encompass much of what you see here. Limb development starts with identification of, with specification of limb fields. And the limb fields are where the limbs have a potential to form. And then later, there is the induction of the limb development, of the limb bud. The limb bud will form, it will start an outgrowth, and at a certain stage, the patterning will begin, the patterning of the limb bud, the axis formation. And then further on, we will have things like cartilage formation, we have digit specification or digit development, and eventually we have the separation of the digits and all of that. So if you remember little else from this talk, try to remember this outline, because this is sort of the general view of what it looks like when a limb develops. So why do we study limb developments? Well, this might seem silly, but it's kind of convenient that limbs are on the outside, because it means that it's very easy to score a phenotype, and they are very easy to manipulate. Now, it's a lot harder if you try to manipulate things like lungs or hearts, as you can imagine. But limbs are quite simple to play with. And what's interesting with them is that they share a general pattern over vertebrates, but there's a lot of species variation happening. Now, the good thing about the manipulations that you can do, whether they be physical or genetical, is that they are generally embryonically non-lethal, which is very convenient. It means that, well, you can study the whole development. The embryo doesn't die because you knocked something out in the wrong place. It's very, very convenient. Right. So let's define a vertebrate limb. We can look at them as paired structures, which sticks out from the body used for locomotion. And here we have a variety of vertebrate limbs. As you see, they share a lot of features, but the outline is the same, but there is a lot of variation. Now they contain a bony frame with joints, which you can see in this picture. And they are covered by muscles, and they are of course innervated. And this is important, they develop from a limb bud. So what is a limb bud? This is one of the other things which I really want you to remember, the limb bud. This is about a 4-day chick embryo, which was serendipitously stained in my in situs for a gene that is expressed in the limb buds. So the limb buds are these beautifully bluish, purplish things that sticks out from the body. And they have a number of features which will appear over and over again in this lecture. They have, to start with, they have an apical ectodermal ridge, the AER. So this is the ectoderm at the distal tip of the limb bud, and they are really important for distal signaling to the rest of the limb during development. We have something called a zone of polarizing agent, which sits in the posterior end of the developing limb bud, the ZPA, and it's really important for posterior signaling, for setting up the anterior posterior axis. And we also have something called the progress zone, and this is essentially where all the cell proliferation is happening in the limb. And the limb bud will get contribution from mesenchyme from the lateral plate, mesoderm. And later during development, you will have myocytes migrating from the hypaxial myotome bud, which is essentially, it's the ventrolateral part of the somites. They will move into the, migrate into the limb bud and form all the muscles. And also everything is covered by an epidermis and an ectoderm. And this is also important for signaling processes during the development. Okay, let's look at general anatomy. We have here is a schematic of it's like a day 2 chick embryo. And we have the gray is the paraxial mesoderm, it's all the somites. We have various versions of lateral plate mesoderm. This is actually a typo. It should say somatic and not somatic. So this is all somatic lateral plate mesoderm. And we have something here called forelimb field, which is where we will develop a limb bud. Now, you remember I talked about axis. A lot of this talk will focus on how we are specifying the various axes of the limb. And of course, as you all know, we have a dorsal and a ventral side when you just look at your own hand, and you have a posterior and an anterior side. Now, the anterior-most digit is the thumb. The thumb is digit number 1. And then the pinky is then the most posterior finger. And this is what it looks like in the limb bud, kind of the same view early on. And of course, because the limb grows distally, we have a proximal to distal axis as well, which we can see here from the body center and outwards as it grows. And the limb itself can be divided in a number of different parts. We call them the stylopod, the zygopod, and an autopod. This is a human forelimb. We can see that they're almost containing a special set of elements. So the stylopod contains the humerus, the zygopod will then contain the ulna and the radius, and the autopod is the hand with the digits and everything. And this is a chicken forelimb, so this is a wing, and this is a horse. And you can see that the elements are all recurring. There may be reductions, but you will never have more than usually 2 bones in the zygopod, and almost never more than 5 digits. Okay, let's jump straight into limb fields. Now, I'm sure you understand that limbs are always formed in the same position. And what determines these positions are limb fields and flank. Flank is the part of the body that is between the limb fields. Now, if we look at the forelimb, the forelimb limb field is set up by a retinoic acid gradient already in the primitive streak. So this happens very, very early. We have, let's see. Yes. So what retinoic acid does is that it activates 3' HOX genes, HOX4 and HOX9 parallel group genes in the lateral plate mesoderm. This gives the anterior trunk identity. We also have OCT4, which represses genes of the more 5' groups like HOX10 and HOX13, HOX10 to 13 genes. These are the genes that gives posterior trunk identity. So the forelimb field suppresses hind limb field identity. And we can, of course, fiddle with this by, let's say, we we have a HOXB4 double negative mutant, and this one will have the forelimb shifted anteriorly. And if we deplete OCT4, what happens is that the flanking region will be reduced, which means that we will get the hind limb bud forming just after the forelimb bud. In the hind limb, we have a question? No? In the hind limb, we have another gene, we have GDF11, which suppresses retinoic acid. So it suppresses anterior trunk identity, and it activates HOX9 to 13 paralog genes. These ones are then the posterior trunk identity. And if we play with this, let's say we prematurely activate GDF11, then we will also get an anterior shift in the limb field. So what's happening is that, again, just like up here, we will have the posterior limb forming just next to the anterior limb. And if you want all the gory detail, do check out the review paper, because there's a lot more data there. Okay, so now we have a limb field. Now we have an area where we can actually form a limb. So what about limb initiation? Well, the initiation of the limb bud is an effect of an antagonism between FGF8 and retinoic acid. If you look at the pictures, you here have a wild type mouse embryo, and we have stained all the retinoic acid activity. So this is the border of retinoic acid activity, and we have FGF8. As you can see, we have it's in the heart field and in the more caudal part. Now let's see. Yes. So retinoic acid will activate TBX5 in the lateral plate. Here is TBX5 in the lateral plate. We'll talk more about TBX5, but TBX5 is important for the forelimb bud development. Okay, so what happens if we block retinoic acid activity? Well, here we blocked it. You see there is no marker gene for retinoic acid. We get an expansion of FGF8, which means that we will have no activation of TBX5 in the lateral plate. This mouse will not have any forelimbs. Now, TBX5 stimulates the lateral plate mesoderm to undergo epithelial to mesenchymal transition. So this is a cross-section of I guess it's a chick embryo at the area of the limb field. We have, this is the central nervous system, the neural tube, we have a somite here, and this is the lateral plate mesoderm. This is the somatic mesoderm, and this is the splanchnic mesoderm. Now what TBX5 does is that it causes these cells to migrate out towards the ectoderm. And in this experiment, they tagged cells in the coelom, so with GFP. So here they are, nice and green, and they are sitting happily in the epithelial mesoderm. And TBX is turned on, and these cells start to migrate out and forms this rather messy-looking thing. So cells will end up in this little area. Okay, what TBX5 also does, it stimulates FGF10 in the mesenchyme, in those cells that just emigrated via the Wnt pathway. And here we can see FGF10. This is an in situ, I guess. So we have it right here. And what we can do is that we can put in a small clump of cells which are making FGF10, and we put them into the flank of the embryo. And then we will get an extra limb. So this is then the outcome if we embed FGF10 cells into the flank. We will have one hind limb, two hind limb, three hind limb, which is kind of weird. But this is a really good readout if your cells has FGF activity. FGF10 in turn, which is now in the mesenchyme, it activates FGF8 in the ectoderm, which leads to the formation of the apical ectodermal ridge, the AER. Now, in the hind limb, we have TBX4 in the chicken or islet1, which has a similar function to TBX5 in the forelimb. TBX5 gives the forelimb identity, while then TBX4 and islet1 give the hind limb identity. And we can, of course, play with this genetically, and we can create hind limb identity in the forelimb and vice versa by modifying these genes. Now, one interesting little experiment you can do is that you can, so this is an embryo, you have the expression of TBX5 in the anterior part, TBX4 in the posterior part. If we put a bead soaked in FGFs in the flank region, it's kind of similar to this experiment. Depending on where you put it, you will get a different result. So if you put it close to the forelimb band, you will get a wing. This extra bud will express TBX5, and you will get a wing. If you put it close to the hind limb bud, it will express TBX4, and you will get an extra leg. But if you are really tricky and you manage to put it just in the middle, you can get a chimeric limb. So it's a limb that has the anterior part as a wing, but the posterior part as a leg. And this is then photos of the actual experiments. Here we put a bead in the flank, FGF bead in the flank, close to the anterior part. You can see that there is TBX5 expression. This one will give you a duplicated forelimb. In this case, you have it in the hind limb sort of area, so it expresses TBX4, and you would get an extra hind limb. So this is the extra wing structure, I guess. Yeah, that looks like an extra wing. And this is the chimeric limb. So it's where we put the bead smack in the middle, and we have the anterior part is the wing, and the posterior part is the leg. Okay, so let's look at the actual model that we have. Retinoic acid activates TBX5 in the lateral plate mesoderm. Here, this is the forelimb. Retinoic acid in the forelimb field activates TBX5. TBX5 leads to epithelial to mesenchymal transformation, and cells migrate out and forms a loose mesenchyme. TBX5 then, via the Wnt pathway, causes the activation of FGF10 in this mesenchyme, which causes cell proliferation and activation of the apical ectodermal ridge. Yes. Right. Now, FGF8 in turns, it stimulates FGF10 back. So you will have a positive feedback loop happening here, which causes the expansion of the limb bud. In the hind limb bud, it's actually pretty much the same, apart from we don't know what the initial signal is because, well, it's not retinoic acid, that we know. But we have TBX4 or islet1, which has similar function as TBX5 has in the forelimb. Okay, let's go into axis formation. Now, the bones are specified and formed in the proximal to distal sequence. That means, that means that we will have the stylopod is formed, well, specified first, and then we have the zygopod, and finally the autopod. Yes. Now, people have been playing with limb development for quite some time. Already in the early 20th century, people did transplantation experiments in amphibians and chicks. And in 1948, a paper was published by John Saunders, where he had been removing the apical ectodermal ridge at different time points. And this was published in 48. I met John Saunders in 2003. That's he had a very long career, I can tell you, very long career. He was a really nice guy. So if you remove the apical ectodermal ridge early, you will only get the formation of the humerus, which is this proximal part. This is an AER, an apical ectodermal ridge in a chick embryo. This is the side view, and this is a view from dorsal. And it's been stained most likely with, well, it looks like FGF8. So it is an actual ridge that sticks out of the bud. So if you remove the apical ectodermal ridge slightly later, you will get the formation of the humerus and the radius and the ulna. So you will get all these elements. You don't get the hand though. So what does this mean? Does this mean that the positional information is coming from the apical ectodermal ridge? Maybe more experiments are needed. So because if you take the apical ectodermal ridge from an old limb bud and you replace it with a young limb bud, or the other way around, you will have no effect. So the age of the limb, the age of the apical ectodermal ridge has no impact on the development of the limb. However, if you put young progress zone cells into an old limb bud, you will get a new limb forming kind of at the distal end of the original limb. And this is what it looks like. It's kind of weird. So here's a limb bud, and they put these progress zone cells from an old limb bud, and they put it into a young limb bud. Now the young limb bud was happily prepared to form first the stylopod and the zygopod, and then we put in these old cells, and these cells again happily produced a new stylopod and a zygopod, and then eventually we got a hand. So yeah, we have, this is one arm, and this is the second arm. And if we do the opposite, if we put old progress zone cells into a young bud, we will get a truncation. And this is what it looks like. So here we took old progress zone cells, that is cells from an old limb bud, and we put this into a young limb bud, we will get this truncation happening. Now you can see that the hand part, it looks pretty normal, but the rest is kind of lacking because all these bones are just truncated. They didn't form properly. Let's do some more experiments. What these experiments tells us is that the apical ectodermal ridge is responsible for the cell proliferation in the mesenchyme. And here's an outline of a number of experiments. This is the John Saunders experiment that I talked about. You remove the AER, the apical ectodermal ridge, and the limb development ceases. If you put an extra apical ectodermal ridge next to the original one, you will get a duplication of the wing. If you put leg mesenchyme, which you put leg mesenchyme cells into a wing bud, you will have first a part of the wing forms, and then you will get a leg on top, which is kind of bizarre. If you put non-limb mesenchyme instead of limb mesenchyme cells into the limb bud, what happens here is that these cells, they cannot signal to the apical ectodermal ridge, which means that the apical ectodermal ridge cease to function and the limb development stops. We also figured out that the apical ectodermal ridge can be replaced with an FGF bead. So here we remove the apical ectodermal ridge and we put an FGF containing bead instead, and you will get a normal wing. So we figured out that FGF is a key signaling factor, and that the mesenchyme contains the positional information and the identity of limb development. And we can do even more experiments. So in these experiments, they cut the limb bud tip at different time points of development, and they put it into the neck of the embryo, which is, it's a kind of a Frankenstein experiment. What you get from this, so this is the tip of a very young limb bud. You put it in the neck and you will get an autopod, pretty much. If you do it with a slightly later limb bud tip, you will get, still you will get the hand, but it's kind of slightly less. And again, even later, you will get a smaller piece of the hand. In this case, it's just a finger. So what it tells us is that the younger the graft, the more proximal elements will develop. However, if we treat this graft with retinoic acid, which is this column, all the elements become more proximal. So this is then the young limb bud transplants. You see that we get pretty much the whole limb here. In the next stage, we get a structure which is kind of similar, more similar to this. So we get parts of the hand, but we also get these, maybe it is the stylopod, the small zygopod, maybe. And finally, we just get more hand structures. We can do the reverse experiments. So instead of treating it with retinoic acid, we take the mesenchyme cells out and we put them in the same part right here, and we'll see what happens. So what happens is that we put the untreated mesenchyme cells, and we look at the proximal genes, and in this case it's mice1. We see that we have mice1 in like half of whatever weird kind of bud structure that grows. HOXA11 is important for the zygopod. That is this part, that's why it's yellow. And finally, we have the distal hand genes, which in this case is HOXA13. And so this is the normal. Now, if we dip this if we incubate it with FGFs and WNTs, we will see that the mice1 is lost. We have a slight expansion of HOXA11, but most of all, we see that the distal genes are highly upregulated. Sorry. If we compare these two. What this tells us is this model. We have retinoic acid in the flank, which induces mice1 and 2 proximally. We have FGFs and WNTs in the distal part, and they will induce HOXA13 and CYP26B1, which degrades retinoic acid and inhibits mice1. Which means that we have two sources of signaling, and they will create three separate domains which are responsible for making the three parts of the limb. This is sort of the current model we're working with. So we have gradients of retinoic acid and mice, and we have gradients of FGFs and WNTs and HOX13. And together they will cause the three elements. So proximal will give rise to the stylopod, intermediate, the zygopod, and distal, the autopod. Okay, now again, let's talk a little bit about HOX genes because it's so much fun. If you are interested in HOX genes, I recommend you to look up Denis Duboul's lab, he's in Geneva in Switzerland. He's done a lot of work on how HOX genes function. Okay, as I mentioned, there is spatial and temporal collinearity of expression. We see this here. What does it mean? It means that the group 9 and 10 elements are important for the stylopod formation. The group 10 and 11 are important for the zygopod elements. And finally, the group 11 to 13 are important for the autopod elements. And yeah, and you see it's not a huge difference between forelimb and hindlimb. And of course, people have been playing with this. So in this case, let's see, yes, we have a normal mouse limb. You can see it has a humerus, a radius, and an ulna, and you have a hand. Now, what they did then was that they knocked out HOX-A11 and HOX-D11, and what they got was this kind of truncated arm. You still have the humerus. The radius and ulna might be these tiny bones, and then you have a hand. So what they did was that by knocking out the HOX11 group genes, you essentially removed the zygopod part of the arm. This is a human mutation in HOX-D13, which causes these horrible truncations of the digits, because HOX-D13 is kind of important for making the hand region. Okay, let's go into the anterior to posterior axis formation. Now, these early experiments I told you about, I mean, these experiments that were done really early, even in during the first half of the 20th century, because they were moving a lot of cells back and forth, and they were fiddling with the external part of the limb buds, they figured out that there was a signaling center in the posterior mesoderm. Then they called it the zone of polarizing activity, which is then the ZPA. And this region it has posterior identity. If we transplant this into the anterior end of a limb bud, you can transform the anterior identity into posterior identity. So this is what they did in this case, and they received this a mirror image duplication of a chicken hand, which is really weird, but fascinating at the same time. Okay, this signal, which is responsible for this zone of polarized activity, is sonic hedgehog. This is another chick embryo, and it's an in situ hybridization for sonic hedgehog, and we can see that it's in the posterior part of the limb buds. So we can do the same experiments as in the previous experiment, but instead of moving cells, we can put in sonic hedgehog producing fibroblasts as a proxy for the zone of polarizing agents, and we will get the same mirror image duplication. This is a normal mouse hand. This is what the Sonic Hedgehog expression looks like in a mouse embryo. These are the limb buds. It's in the sort of posterior part. If we overexpress Sonic Hedgehog in almost the entire limb bud, like in this case, we would get polydactyly. We would just get a huge number of digits forming. And this happens in humans. Here you have a polydactylous human limb, and it happens naturally in other animals. I mean, well, it happens naturally in humans too. There is a population of polydactylous cats. So the thing is, Ernest Hemingway, the author, he was really into polydactylous cats, which might sound like a weird thing, but well, you know, authors. So he bred a lot of these cats, and these cats are still around on his, I guess, in his house, which is it's in Florida Keys. It's a group of islands south of Florida. So you can still find them, the descendants of the cats that he collected, and they're all polydactylous. It's really weird. Okay. The digits are formed in a spatio-temporal specification, most likely due to Sonic Hedgehog signaling. Okay, so we have we get a Sonic Hedgehog gradient set up in the limb bud. We have a question? No?
Rolf, I have a question.
Yep.
So this is Niveta. Hi. And
yep.
You spoke of polydactyly occurring naturally. So
yes.
I was wondering if that is also because of natural overexpression of Sonic Hedgehog.
Yeah, well, there are actually many reasons for polydactyly. And one of them is that you have a natural overexpression, or you have an expansion of Sonic Hedgehog expression in the limb bud. Yes. But there are other genes which causes polydactyly. I should tell you that.
Yeah.
Yep.
Okay.
I guess in this case, because they are showing this picture, I guess it's because of Sonic Hedgehog overexpression. But you know, from the outside morphology, you can't really tell if it's Sonic Hedgehog or if it's GLY3 or a combination. But there are many reasons for polydactyly.
And since Sonic Hedgehog is a molecule involved in a lot of signaling pathways and a lot of developmental processes, so it's just weird to think that if Sonic Hedgehog overexpression can occur naturally, whether it will have other phenotypic defects as well.
I will actually give hints to that question later in this lecture. It's about Sonic Hedgehog control. There are enhancers. I'll show you.
Okay, thank you.
Yes. So, more on Sonic. Yes, where was I? There's a Sonic Hedgehog gradient set up in the limb bud. We have high concentration in the posterior part, and gradually less the more anterior we get. And the first digit 1, the thumb, it's specified first, and it's specified in an area which has never seen or experienced Sonic Hedgehog. The second digit forms about 4 hours, well, I should say specifies 4 hours later in the low Sonic Hedgehog concentration area. And then digit 3, which specifies 8 hours later, has then sort of a medium expression of Sonic Hedgehog. And finally, digit 4 and 5 are specified 12 hours later. And these digits are descendants of cells which originally expressed Sonic Hedgehog. The difference between digit 4 and 5 is that digit 5 has spent longer time in a high Sonic Hedgehog concentration area. So this is the experiment. They tagged cells in the zone of polarizing activity really close to each other. And you see that these cells separate. So originally they are part of the zone of polarizing activity. Then they get separated and they form different digits. So digit 4 has, the cells of digit 4 has spent less time in a high concentration area than the cells of digit 5. And if we knock out sonic hedgehog completely, we will get a very poorly developed digit 1. So it's it's the opposite of the polydactyly. Okay, now since we haven't talked about BMPs much yet, let's do that, because they are important for digit identity. Okay, so digit identity, apart from sonic expression, it's also specified by the interdigital space. So these are then the digit unligands, and this is the area which will form the digits later during development. If we remove this part, the red part here, between digit 2 and 3, what happens is that the identity of digit 2 gets transformed into digit 1, which kind of makes sense, because what you're doing is that you're removing sonic hedgehog from this area. If you do the same of the interdigital space between digit 3 and 4, you will again get a shift in identity. You will have a shift from identity 3 to identity 2. And this can be mimicked by placing a noggin bead. Now, noggin is a BMP inhibitor. So you put a noggin bead, you block BMP, and what happens is that, again, you have the same transformation of identity from digit 2 to digit 1. If you put it between digit 4 and 3, what should have been digit 3 now becomes digit 2. We'll get back to this. The model. So, as you remember, we have FGF10. It initiates the apical ectodermal ridge via WNTs. And early during development, if we look down here, we have sonic hedgehog in the zone of polarizing activity. It activates gremlin. Gremlin stimulates FGF8 in the apical ectodermal ridge by inhibiting BMP4. The FGFs in the apical ectodermal ridge stimulate sonic hedgehog in the zone of polarizing activity. So here we have a positive loop. If we look slightly later during development, we have limb bodies looking like this. The high levels of fibroblast growth factors starts inhibiting gremlin. If we inhibit gremlin, then we have BMP4s, which will start repressing the FGFs in the apical ectodermal ridge. If we look even later, then the BMP levels have gotten high enough that they are entirely blocking the FGF production in the apical ectodermal ridge. So the apical ectodermal ridge disappears. This causes also the loss of the zone of polarizing activity. So this is the end of all the signaling that is happening in the limb bud. From this stage, we will have just mainly cell proliferation happening. And this is another view of the model. Sonic hedgehog activates gremlin, inhibits BMP, gremlin activates FGF8. FGF8, when during early development, activates sonic hedgehog. During later development, FGF8 will block gremlin. We will have high levels of BMPs, which will block the FGFs, which will cause the end of the apical ectodermal ridge, which in turn would cause the end of the zone of polarizing activity. Right, let's go back to digit identity, and let's go back to the Hox genes. You may not have heard this, but there are two phases of Hox gene expression, well, especially in the limb. The first phase, in the first phase, we have the early limb control regulatory region, the ELCR. This is the Hox operon, and it will initiate Hox expression from the 3' end. So expression will start here and it will continue downwards. And it also activates the posterior restriction region. Now, this imposes a spatial restriction on the 5' Hox genes. So it would cause a nested expression pattern. And that's what you see here. So during the young limb bud, these are then the expression. I should say this is the anterior borders of these Hox genes. So Hox D10 is actually expressed in the whole limb bud. Hox D11 is expressed in the limb bud up to this point. So in the posterior part, up to that point. D12 is expressed in this part, and D13 in the most posterior part. So this means that in this posterior part, you have all these Hox genes are expressed, while in the anterior part, you only have like D10. And then slightly later in development, what happens is that the Hox genes will activate the long-range enhancer of Sonic Hedgehog, which starts Sonic Hedgehog activity in the zone of polarizing activity. And people have been playing with this enhancer and they have been dissecting it. So what they can tell is that this enhancer specifically drives a Sonic Hedgehog expression in the zone of polarizing activity. So this kind of answers your question to how this works. It's because we have a tissue-specific enhancer for this little spot for Sonic Hedgehog expression,
Yeah,
which is pretty cool. And this is like I think is 1 megabasis upstream of Sonic Hedgehog, so it's really far away. Okay. Together with Hox B8, Sonic Hedgehog is activated and restricted to this zone of polarizing activity in the posterior limb bud. Now the second phase, Sonic Hedgehog now activates the global control region of the Hox cluster, which is down here. Now this causes an inversion of the expression pattern of the 5' Hox genes. So if we look here, this is the later limb bud. We notice that D13, which was early, it was restricted to the posterior region, is now the anterior-most Hox gene to be expressed, which is kind of weird. And yeah, this means that we would get this nested pattern, and the first wave is responsible for making the stylopod and the zygopod, and the second wave is responsible for the development of the hand or the autopod. And please try to remember this for next lecture on Wednesday, because I might talk about this when I talk about evolution of limbs. And so this pattern, the collinearity of Hox genes and where the enhancers and everything and how they work, this was worked out by Denis Duboult's lab. So he has published a lot on Hox gene expression, Hox gene development, and Hox gene function. Okay, one more axis. Experiments that were done for like 56 years ago, where they rotated just the ectoderm of the limb bud, it caused a reversal of the dorsal to ventral axis. So yeah, which is interesting. What people found out later is that there is something in the ectoderm. There is Wnt7a in the dorsal ectoderm. And if you knock out Wnt7a in a mouse, it causes the ventralization of the dorsal side. It means that the dorsal part of the mouse will, the mouse limb will look like the ventral, and you will no longer have a dorsal side. And also they figured out that you have LMX1B expression in the dorsal mesenchyme. And if we lose this expression, we will get, again, a ventralization of the dorsal side. And I'll show you what it looks like, because it's just weird. Okay, so here is a normal mouse limb. And the cross section, we can see that has these little foot pads. We can see them in the cross section. These are the digits. Here they knocked out LMX1B, and as you see, that something is seriously wrong here. You have the foot pads on both the ventral and the dorsal side, which we also can see in the cross section. So the dorsal side just got ventralized. It also means that there will be problem with innervation and joint formation. Yes. And if you overexpress LMX1B in the ventral mesenchyme, so this part is usually LMX1B blank, but if you overexpress it into the whole limb bud, you will get a dorsalization of the limb bud. So instead of this feature, you will get kind of the opposites. You will have no foot pads anywhere, and yeah, it will just look strange. Okay, there is more stuff happening. Again, there are BMPs, and you have BMPs in the ventral mesenchyme, and the BMP signaling will induce engrailed-1 in the ventral ectoderm. And of course, we can play with this. If we block BMP, we cause an expansion of the Wnt7a into the whole limb bud ectoderm, and the limb gets dorsalized. Now, you might think that, yeah, all these axes, they are nice and separate, and that's all. But no, it's all connected. Now, this Wnt7a mutant, it also lacks posterior digits. And these embryos have a reduced sonic hedgehog activity, because it turns out that Wnt7a, together with FGF4, stimulates sonic hedgehog activity. So if you overexpress Wnt in the ventral ectoderm, you get an enlarged apical ectodermal ridge and extra digits. Huh. BMPs are then involved in modulating the proximal to distal axis and digit identity and the dorsoventral axis formation. Same molecules, all the different axes. Now, Hox genes are then responsible for the limb field positioning, the very, very early stages, the proximal to distal development and identity of the elements in the proximal to distal developments, and also the anterior to posterior development and identity in the hand part. And I could do the same for the FGFs. What I want you to see is that it's all connected. You cannot remove one axis without affecting the development of the other axis. So let's go back to the overview. This is limb development. This is what I have been talking about today. We started out with the limb field specification, which is actually not in this slide because The idea of limb fields is more modern than this picture. Anyway, we have the limb field specified first, then we have the induction of the limb. And as you remember, we had retinoic acid, and we had OXIS, and we had TBX5 in the forelimb, and we had TBX4 in the hind limb. And these genes specify the identity of the limbs. And then we will have FGF10, which will cause cells to proliferate, and it induces the apical ectodermal ridge. And when we get the apical ectodermal ridge, we get even more proliferation, and we get the onset of a zone of polarizing activity in the posterior part, which starts patterning the anterior to posterior axis, and is also a major player in the proximal to distal axis. And yeah, we didn't really talk about chondrogenesis, but maybe you remember that I said that digit specification happens in a spatiotemporal pattern. However, digit development doesn't happen in the same order as the digit are specified, just for your information. Yeah, digits form, they get the identity, and voila, you have a fully functional limb. So I just scratched the surface. There is tons and tons of information. People have gone into various details about various parts of these processes. And if you are interested, do dig out the review that I showed you earlier. I think Vasha is going to upload this talk to your Moodle accounts. If you have any questions, please ask them. We'll see each other again on Wednesday when I'll talk about the evolution, where limbs come from, and in some way how they get that shape. So, any questions? My soothing voice have lulled you all back to sleep. I appreciate that. Well, if you have no questions, I'd like to thank you for the attention, and I hope to see you all back on Wednesday. And I guess Wednesday is the last lecture, or has Raj put in anything extra after this?
No, there is a lecture. There is the final lecture on 30th of December, where Raj will talk about head development.
Ah, yeah.
And that would be the end of the class. Yes.
Cool. It's good that I showed you the Hox genes before the head lecture, because they will reoccur in a massive way during head development. You can do all kinds of weird stuff in the head by blocking Hox genes. Okay, well, if no questions, I'll see you back again on Wednesday. And have a nice week until then. Thank you.
Thank you, Rolf.
Thank you, Rolf.
Thank you.
Thank you.
Thank you.